At this point, I want to hand it over to Len to tell you more about SIVA and specifically the therapy being developed, targeted hyperthermia therapy, and how it uses Sona's biocompatible gold nanorods. Len.
Well, the concept of targeted hyperthermia is really very simple. As the name implies, it targets hyperthermia, high temperature heat, to solid tumors. And heat does a number of good things for tumors. And as we mentioned earlier, the therapy relies critically on gold nanorods as part of the treatment. And so having those biocompatible gold nanorods really is part of this unlocking of the application. Next slide, please. So the way we've approached this is to look, first of all, at current cancer treatments, which, as everyone knows, have risks, have side effects, they're increasingly expensive, and people really aren't getting the outcomes that we would hope for. Now, cancer is a very challenging problem, and of course, it's going to be just as challenging for our approach as for any other. But what we can do critically with targeted hyperthermia is target gentle therapeutic heat directly to the tumors. What gentle therapeutic heat does is 4 things that we know about that are very beneficial for tumors, and we'll go more into that in a moment. But basically, the way it works is that the patient goes into the clinic one day, they get an intravenous injection of gold nanorods, they go home, they come back the next day, and they get a 10-minute treatment with infrared light for the region that has the tumor or tumors. The gold nanorods are concentrated in tumors. They absorb that infrared light. As David mentioned, the rods can be tuned to different wavelengths, and they emit very gentle therapeutic heat that emanates from within the tumor mass. And so this is work that really can be made commercially practical now because of large-scale production of gold nanorods. Next, please. So the system, as we mentioned, is a 2-component medical device system consisting of the gold nanorods and then also of an infrared light device. Now, this is infrared light. It's basically a fancy heat lamp. It's not radiation. It's not ionizing radiation. And the way they work is, as we mentioned, the nanorods absorb infrared light, which passes through tissue very efficiently, and they convert that into heat. This heat, this gentle heat, does 4 things that we know about. It stimulates the immune system, it shrinks tumors and kills cancer cells, it increases perfusion of the tumor so drugs can do their job better, and it also takes out stem cells, cancer stem cells. And so a number of good responses that you get. Tumors have leaky vasculature, and that's been known for many, many years, and this is called the enhanced permeability and retention effect, or EPR effect. Because of that, material of the right size, the right shape, and so forth can basically leak into tumors through their capillaries faster than it leaks back out, because the capillaries have to remodel and grow as the tumor is growing. With that, we see a 7-fold concentration of material, of nanorods in tumor versus non-tumor tissue. And that differential is one of the factors that gives us enough specificity to heat the tumor directly. Now, healthy cells can withstand high temperatures. Tumor cells are much more sensitive to high temperatures. And so what we want to do is not ablation, which is very high temperature that cooks tissue, but this gentle hyperthermia where we get into the zone, the temperature zone that cancer cells are sensitive to, but healthy cells are not. And this is because of the production of heat shock proteins that are stimulated by that heat. Cancer cells are more sensitive to these, and they go into a cycle of programmed cell death. So it's a very good selective mechanism that takes advantage of a natural property of cancer and cancer cells specifically. Next slide, please. When we talk about hyperthermia versus ablation, there's a big difference. They're both heat, yes, but ablation is high temperature that basically cooks tissue 55 Celsius and above. And so just to give you an idea, if you think about cooking a steak, this would be medium to medium well done steak at the lower end of that temperature range. Hyperthermia, on the other hand, is 44 degrees Celsius, which is cooler than what's commonly called blue rare for meat. So it's not cooked. We don't want to cook the tissue. We want to warm it up to this gentle temperature, a little higher than a high fever, but really not that much. And this is where these important things happen: shrinkage of the tumor, increase of circulation, stimulation of the immune system, and so forth. Next slide, please. When we talk about targeting, this is also important, and it's something we can do uniquely because of the gold nanorods. Now, there are other approaches for getting heat into the body, but there are several main limitations. First is that they're virtually all for ablation, these very high temperatures. Ablation works, but it's not selective. Second is that they're directing energy in from the outside of the body, and so healthy tissue is damaged on the way into the tumor. Now, with gold nanorods, which is shown on the right side of this slide, the patient is injected, the nanorods concentrate in the tumor, infrared light passes through tissue harmlessly until it reaches the nanorods. They warm up the nanorods, and therefore we can heat the tumor from the inside out. This is a big, big, big advantage. We're basically heating inside out rather than outside in, which is what we want to do with tumors. Next slide, please. Here's a snapshot of some animal data. We have a lot of preclinical data already. On the left shows a study in which we used targeted hyperthermia on a rodent tumor model. And you can see that there's 4 lines there. So we did all of the controls. We had infrared light with saline instead of nanorods. We had nanorods without infrared light, and so forth. The green line, however, is the interesting one. That's where we had the combination of infrared light and gold nanorods in the tumor in the mice. And there's dramatic shrinkage of the tumor in that case. On the right is another study in which we looked at the effect of both the drug of choice for a rodent tumor model and targeted hyperthermia. And in this case, the results were really dramatic. So day 0 on the left shows the animal right after treatment. It's a large multi-lobe tumor, and you can see only 19 days out, the tumor mass is gone. That's the same animal in that picture. A little bit of scabbing, but that scabbing healed, and the animal was healthy, gained weight. We took this study with these animals out to 95 days, and 2/3, 66% of the animals that were treated with targeted hyperthermia were alive, healthy, gaining weight, and so forth at 95 days. We sacrificed those animals, and there was no evidence of tumor histologically. An excellent result. In contrast, with the drug of choice for this tumor model, all of the animals were down by 28 days. So a dramatic improvement over drug of choice. Our rodent studies indicate that hyperthermia is doing a better job for these tumor types than the drug. The combination of drug and hyperthermia is the best of all, which is what we might expect. So what we're planning next is to do a large animal study in a swine model for colorectal cancer. Yeah, next, please. That's good. So this is the initial therapeutic application we're going after. When you choose one of these, you have to look at a number of factors. What's the market size? What's the unmet need? What is the clinical workflow that we have to integrate into? If physicians are not going to use the technology in their clinical workflow, well, then it won't be adopted. What is patient response to this treatment? Since our treatment is extremely safe, it's minimally invasive, it doesn't have the side effects of chemotherapy or radiation or surgery, we've seen so far a very, very positive response from the patient advocacy groups we've talked to. So it's really a good area. In addition, there is a large and available population of patients for clinical studies, which is critically important. So our plan is to go into a window where currently patients are typically treated with a degree of drug therapy and a degree of radiation therapy. That is primarily to delay the need for surgery. And the reason we delay the need for surgery is that the results of surgery are devastating for lifestyle. This is specifically for rectal surgery. It's not a good thing to have. Now, we're quite confident that by getting in earlier, because we have such a good safety profile, at the very least, we can further delay the need for surgery. We anticipate that we will ultimately eliminate the need for surgery in these patients and give them a much better outcome than they would have otherwise. So we think it's a really good, suitable initial indication. It's a good market size. There's a team of doctors who like this kind of work and are willing to use it in their workflow, and there are patients who are really eager to get this sort of treatment. Next, please.